A new Oregon State University study has found that the gelatin in the skin of Pacific whiting, an abundant fish on the Pacific coast of North America, may help prevent skin wrinkling caused by ultraviolet radiation. Pacific whiting is caught in large volumes in the United States, but consumers have little familiarity with the mild white meat fish, also known as hake. It is popular in Europe though, where it is the 8th most consumed species. In the US, the 10 most consumed species account for 77% of total per capita seafood consumption, and Pacific whiting is not among the top 10. By studying Pacific whiting, Jung Kwon, an assistant professor at Oregon State's Seafood Research and Education Center in Astoria, Oregon, is looking to change that and elevate pressure on stocks of those 10 species, which include salmon and tuna. She studies marine organisms and their potential to improve human health and is particularly interested in the benefits from parts of marine organisms such as fish skin, which many US consumers choose to discard rather than eat. Fish skins are an abundant resource that we already know have valuable nutritional properties, but the researchers wanted to find out what additional potential value might be found in something traditionally considered a byproduct. In a paper recently published in the journal Marine Drugs, Kwon and a team of researchers looked at molecular pathways that contribute on a cellular level to the wrinkling of skin. That wrinkling is promoted by chronic exposure to ultraviolet light, which breaks down collagen in the skin. The researchers extracted gelatin from Pacific whiting fish and then looked at what impact it had on antioxidant and inflammatory responses and pathways known to degrade collagen and promote synthesis of collagen. They found that the Pacific whiting skin reactivated to a certain level the collagen synthesis pathway that had been suppressed by UV radiation, and prevented activation to a certain level of the collagen degradation pathway that had been accelerated by UV radiation. Also promoted additional antioxidant activity. Antioxidants are substances that can prevent or slow damage to cells, and promoted additional anti-inflammatory effects. Kwon cautioned that these are initial results obtained in her lab through a human cell model system. Further research is needed using animal models. The research was funded by Pacific Seafood, a harvester, processor, and distributor of seafood.